Good morning. Welcome to our Companion Diagnostics webinar, which is an introduction to an event coming up soon, and we're going to learn about a little more about that in a moment. My name is Joanne Geer. Whoops, just went to the end of my slideshow. Very good. I will be sharing with you a little bit about our organization, the Biopharma Research Council. We are a nonprofit association for scientists, engineers, and IT professionals across all the many silos of biomedical research. Because of the data revolution, people can speak to each other throughout all the different silos at this point, and so we have developed a series of programs and activities that include physical symposium conferences as well as virtual programs like today and even virtual conferences. And this is just a small sampling, and these are all, we're returning back to all of these as well. So we did the Internet of Medical Things, which was a rather hair-curling day full of information about cybersecurity for connected devices. We did a program on point-of-care diagnostics. Just last week we had our microbiome update. And earlier this week in Durham, North Carolina, we had D3D, Data Drugs Diagnostics, at the North Carolina Biotechnology Center. This was our 5th year going back for that program. Again, you're going to hear more about companion diagnostics in a moment, and then in November we're doing a manufacturing expo that will really look at the complexities of biologics, small molecule, continuous processing, single-use processing, all the different engineering aspects of the drug development, the handshake between drug development and manufacturing, as well as regulatory issues, project management issues, change management issues, and so on. You can enter questions in the box at the right at any time. I'll be monitoring them, and if it's something that can be addressed immediately, it will be. Otherwise, we'll take all the questions after both of our presenters have spoken. I'm going to introduce now Tom Fair, who is our Director for Strategic Alliances. Tom?
Thank you. Good morning and welcome everyone to the webinar. Before I introduce the speakers, I'd like to talk a little bit about the upcoming Companion Diagnostic event. The Companion Diagnostic Symposium will be held in Edison, New Jersey at Pines Manor on the 2nd and 3rd of November. And the goal or the focus of this event is that companion diagnostics has reached a benchmark as far as acceptance. One can go to the FDA and see a list there of the approved diagnostic devices. And what we've done with this event is get into more how the partnerships and collaborations that resulted in those approvals have taken a key role. So things have moved from a technology-based to a more partnership-based approach. We will cover technologies and biomarkers, of course, in companion diagnostics, but the goal here will focus on forming partnerships, coordinating projects among teams, and the steps required to launch the companion drug and diagnostic. So with that, let me mention we have a great list of speakers, and we have a great set of sponsors for the event, so please come and see that. And among our sponsors is Cancer Genetics Incorporated, CGI. Our 2 speakers today are from Cancer Genetics, and what I'd like to do now is introduce them both. Kamala Madali has a DVM and PhD in pharmacology from University of Missouri, Columbia, and the DVM in veterinary medicine from Acharya NG Ranga Agricultural University in India. She is the Vice President of Collaborations, Biopharma Collaborations and Companion Diagnostics, and has a pivotal role in biomarker and companion diagnostic strategy, especially regarding pharma novel drug development programs. She has over 10 years experience in global P&L and scientific management. Her full bio is available at the Companion Diagnostics 2016 Symposium website. Please go have a look there for more details. Our second speaker will be Dr. Rita Shaknovich. She has a MD and PhD from the Medical Scientist training program in Mount Sinai School of Medicine in New York. She completed clinical training in anatomic pathology, specializing in hematopathology, and finished her postdoctoral training in the lab of Dr. Ari Melnick at the Weill Cornell Medical College. Dr. Shaknovich has over 10 years of experience in pathology, hematopathology, and surgical pathology. She's a physician scientist with clinical experience in these areas and extensive experience in translational research and experimental therapeutics. The focus of her research has been in epigenetic mechanisms governing normal B cell development and lymphogenesis. And I welcome them both today, and with that, I will turn the floor over to them. Thank you.
Thank you, Tom. Thank you, everyone, for taking time to attend our webinar today. As you see on the slide, the topic of the webinar is translating cancer research into targeted therapeutics. And the agenda will be focused on two key topics, primarily cancer
Excuse me, excuse me, dear. I'm so sorry to interrupt, but could you make your slide beautiful? Thank you.
Okay. Yep.
Terrific.
Thank you. Thank you, Joanne. Yeah. The agenda will be focused on two topics. One is cancer, and primarily it is cancer and immune surveillance and practical aspects of biomarker testing. So as you see on the slide here, biomarker trials for patient selection have significantly shown higher probability of success. As you see on the graph on the right-hand side, which has been displayed across multiple therapeutic areas, we have highlighted how oncology specifically has proven a notably higher success rate in phase 2 in the past 5 years, spanning across hundreds of compounds in 500 plus oncology trials. And as they're color-coded, 67% of these were biomarker trials. And in this era of precision and increasingly personalized therapy, the healthcare industry clearly demands cost-effective options that can robustly identify biomarkers to help select cancer patients, and which is in a way for most likely to primarily benefit patients, especially through introduction of novel targeted therapeutics, and also lately, as we all came across, the era of immuno-oncology drugs, where it is touching the lives of every person that we know who is battling with cancer, or friends or family members. So the success of using select biomarkers in these oncology trials, it can be incorporated as patient selection markers, stratification criteria for inclusion or exclusion, arm selection if they need to move on from one arm to the other during the course of the study, and also monitoring, which has led over to the success rate of biomarkers pretty much on its way to even replace survival in some of the cases, because you cannot really carry over the trial for 10 plus years, wherein there are patients who are desperately waiting for actually the drug to be accessible. So biomarkers have, in general, overall, have made a tremendous impact on the overall application and adoption of the drugs early in a fast track or accelerated process lately. The slide here, actually, I want to focus and take a little bit of time, focus about the immuno-oncology evolution that just started in 2015, and how this evolution is going to really touch everyone's lives who is battling with cancer, and every one of us who is committed very closely to the community to make a big difference in a patient's life. So through the course of 1 decade, immuno-oncology drugs by themselves have been shown and also will be proven, as we see more and more data, to be highly effective in 20 to 30% of patients, as you see across multiple targets, very much familiar for many of us who are on this webinar today, PD-1, PD-L1, CTLA-4, and also many other targets like CIT3, and all of these targets and combinations are also very key in this arena. wherein in order to develop more effective treatments with fewer side effects, the immuno-oncology biomarkers and tests are very key in helping us to assess the effective therapies, because these therapies will play a key role, as we have seen very recently through the announcements of the data at ESMO just last week through BMS, Abdiva, and Merck Keytruda programs, which are very notably rewarding programs that have touched really already some of the patients' lives in this arena. And immuno-oncology is a very highly promising area of medicine, and has already we have seen, as I indicated, through few of the companies who have introduced drugs particularly in melanoma and non-small cell lung cancer. Several of these drugs require identification of patients that have a high likelihood of response, which is very key. As a result, healthcare professionals require identification and potentially companion diagnostics to facilitate patient care. Clinical trials and targeted therapies that are under development, which are touching across all these drug targets that are displayed on this slide, and many existing approved oncology drugs are undergoing combination and also monotherapy clinical research to identify patient groups that can benefit from stimulating an effective immune response against cancer. Overall, the goal for us is, through these breakthrough therapies, to achieve a more durable or more effective response to the therapy. And it is very key, as we are seeing in this era of immuno-oncology, tremendous amount of combination trials. Pretty much almost every company has an immuno-oncology drug in the pipeline. It is very key that companies like CGI, precisely who actually are responsible for the study execution and also the value proposition aspects, which is by supporting biomarker strategies and companion diagnostic strategies in relation to these drug development programs that are designed in these trials. It is very key that three pillars of success are key for being an effective partner for design, for execution, and successful adoption of the drug and also their companion diagnostic or complementary diagnostic into the patient care. So CGI, referring closely about CGI, where we represent, we are more than what we perform just on tests. We are focused on quality, patients, and industry. We are an oncology diagnostic partner. Our model is from bench to bedside, focusing on research, partnering with leading research institutes. Our goal is to drive innovation and develop new insights. As Dr. Shaknovich will carry on the rest of the presentation after a few slides, she will be touching on few case studies wherein it is very key, in addition to understanding the pharmacodynamic aspects of the drug, it is very critical to understand the disease-specific insights and have an integrated approach. And with these insights, we actually provide unparalleled expertise to pharma companies, biotech companies, and also diagnostic companies for improved therapeutic development. And in the continuum of the chain of the cycle for companion diagnostics, it is very, very critical for companies like CGI and business models like CGI, who are very committed to precision medicine efforts for delivering critical genomic insights to physicians, and also not only genomic but integrated insights, all the biomarker insights in addition to the companion and complementary and also the biomarker insights to personalize treatment and improve outcomes. Just touching base a little bit on the companion diagnostic aspects, precisely in the context of immuno-oncology, as we have seen that several drugs were approved very recently by FDA with a companion diagnostic component and also a complementary diagnostic component. It is actually very critical that partners like CGI should offer these commercial assays, which we are, especially precisely in the context of immuno-oncology. Several targets are being designed for PD-1 and PD-L1, which are currently FDA approved and also which are also investigational clinical trials. And you will see in the rest of the presentation how critical it is that 5 pillars, which is the regulatory, IT infrastructure, scientific, medical, and technical insights are very key. And CGI has the partnerships established with several diagnostic companies, wherein we play a role as not only as a deployment partner for supporting the companion diagnostic trials in tandem with the drug trials, but also as a development partner with some of our proprietary LDT panels in lymphoma, multiple myeloma, and renal cell carcinoma. So CGI is continuing and also will continue expanding overall capabilities in immuno-oncology and immunotherapy and integrate these capabilities with genomic and biomarker-based testing insights in our Center of Excellences in both New Jersey, Los Angeles, China, India, and also North Carolina. I will pass on the rest of the presentation from now on to Dr. Rita Shaknovich, who will be transitioning the presentation from me. Thank you.
Thank you very much. Good morning, everyone. So I will continue the theme that Kamala started. I will, again, try to focus on the more practical aspects of biomarker testing and also developing of the biomarker-centric tests. But again, I just wanted to emphasize how I, as a practicing pathologist, and I assume that there are other practicing pathologists in the audience, how do we envision what the role of the such companies like CGI or CROs is in the biomarker field. And the key, I think, that I wanted to emphasize is that the advantage being that being in the company at the CRO gives practicing pathologists, for example, is the quick development of the test and also very quick utilization of the new scientific discoveries and taking that from the stage of the innovation to the clinical service, clinical practice, where it actually can serve not only the patients, but also supporting the clinical trials for biopharma. So where do such insights come? So how do we decide which tests to develop? And again, it is a relevant question not just for CGI, for other CROs, but also for academia. Of course, the speed with which certain scientific discoveries penetrate, the translational medicine increase significantly. It does happen very fast. The lag becomes shorter and shorter, but of course, the published literature, accessibility to the information, presentations at the conferences gives us glimpses at what is next and what is upcoming potential novel drug or the novel biomarker. We do also have our in-house R&D department where we do certain research and have a lot of collaborations, and I just wanted to emphasize the practical aspect and the value of the scientific academic collaborations that one can have and benefit from the early research that comes off the benches of the great research institutions, and we do collaborate with such places like Memorial Sloan Kettering, USC, LIJ, Cornell, etc. So I think it's a very valuable aspect. I want to sort of set the stage for immuno-oncology. Of course, there is an explosion of drugs, of understanding, science behind it. The field, of course, is an old field. We've known about the role of the immune system in cancer responses, but I think only now we are able to really harness that knowledge and implement it and harness it for the improvement of the patient's outcomes and fighting cancer. So just wanted, in order to set up the stage, I wanted to use this panel from the paper, with the review paper, which actually summarizes very well various aspects of immuno-oncology. So again, as I pointed out, and as we all know, immune system has a role not only in monitoring and defending our body from various sort of benign non-neoplastic offenses like bacterial viral infections, allergens, but also has a role in controlling cancer, the tumors that grow in our bodies. And so that is based on the ability of the immune system, T cells, B cells, antigen-presenting cells, to recognize the tumor and to destroy it and to fight it, but to recognize it as something sort of foreign. And of course, the whole field and the number of specialized T and B cells is dramatic and growing by the hour because, again, our knowledge and understanding of how T cells are regulated and how we respond to certain antigens is also very much in development. And so when we think about immune system and its response to tumors, one possible way of looking at it is to look at some tumors, and we know that looking at the slide, that some tumors are very T cell or immune cell poor, and this is represented by this image in the table on the left. So there are very few infiltrating T or B cells, macrophages within those tumors. So those tumors have pretty much do not really incite any immune response from the body. They're very neutral. So in order to really address and in order to utilize immune response towards those tumors, the approach would be to activate the immune system. And so, again, there are various reasons why a tumor would be sort of neutral or T-cell poor, and few examples are listed here as basically the immune system is just anergic. There are maybe some immune compromise, and so we know about certain tumors arising in the setting of the immune incompetence, right? About in the setting of HIV or in the setting of some inherited disorders. It's lack of chemokines, so tumors that do not really secrete chemokines that would attract the immune cells towards it. Then stroma preventing cells from penetration towards tumor, large size of tumor that also may play a role, etc. So in this case, one possibility of actually fighting the tumor would be to activate the immune system. And so there are agonistic antibodies. If we look at this image on the left around the T cells, there is a whole slew of molecules that actually are considered to be activating. So if we do use the antibody that is agonistic, that would affect those cell surface markers like CD28 or GITR or CD27, it is possible to actually reactivate certain subsets of T cells, and then in the hope that then they would be more active in recognizing the tumor and fighting it. On the other hand, there are also tumors that are very T-cell rich, and they are, as you can see in the image on the right, they are very densely infiltrated by T cells and B cells and macrophages. In those tumors, so there is a milieu that allows or provokes the very brisk immune response, but at the same time, it could be due to, for example, certain chemokines. We know in Hodgkin lymphoma there is a very rich chemokine secretion, and so that attracts a lot of immune cells. But tumor still finds the way of fighting the immune response, and one way of doing it is actually by, for example, presenting certain blocking antibodies or blocking epitopes like PD-L1 or PD-1. And so there are certain inhibitory ways the tumor can actually suppress the immune response towards it. It can mask itself. So one subset of tumors that is known to attract a very active, brisk immune response is tumor with genomic instability with lots of high mutational load and many neoantigens. We know that, and we're learning how to measure that. But there are ways also through the very quick tumor evolution, the tumor tries to mask those neoantigens and sort of suppress the immune response, as I pointed out. So T regulatory cells are often rich. T regulatory cells also have the ability to finally tune and inhibit the immune response. Some cytokines like IL-10s can very actively suppress the immune response also, or de-energize the immune milieu that is within the tumor. And there are antibodies that are designed in our therapeutic molecules that are designed to target exactly those epitopes that permit immune evasion by the tumor. And we know a lot about CTLA-4 and PD-1, PD-L1 targeting, among others, TIM-3, etc. So that is really the simplified version of how tumors can really evade the immune response and how various therapeutic molecules can actually interfere and reverse that process. And so I wanted to use, of course, the PD-1, PD-L1 example as a case study because there are several drugs that are directed at that checkpoint, at that axis, and those drugs are quite effective and actually improve life of patients with different types of cancers. And also because so much happened in the last several years in that area of PD-1, PD-L1 biomarker and other biomarkers where the whole concept of companion diagnostics finally penetrated the field of immunohistochemistry. We knew about companion diagnostics in molecular testing, but this is something that is very quickly developing and sort of attempts to bring immunohistochemistry from semi-quantitative, sometimes poorly reproducible technique to something that is much more precise and much more quantitative, or at least semi-quantitative. So again, few words about PD-1, PD-L1, and there is a lot of literature, so I just want to emphasize that PD-1, PD-L1 represents just one axis of suppressing the overactive immune response. Normally in the organism it's utilized for often in the setting of, let's say, infection to prevent the excessive response of T cells to the infectious agent and preventing the autoimmune diseases or preventing the immune cells from attacking the normal cells. And so what happens is that there is interaction between PD-1, which is programmed death 1, it's a receptor, with PD-L1 ligand, recognized and usually on antigen-presenting cells. That interaction allows sort of dampening the immune response by T cells. The same mechanisms that are used, that are present in the normal physiological processes are also very effectively used by tumors. So ability to modulate the immune response has been also utilized by the tumor, and we know now so much about it. So we know that those tumors that arise from normal cells that normally would not express PD-1 or PD-L1 actually in the process of the neoplastic transformation upregulate certain mechanisms that just improves their survival. So it is sort of the expedited evolutionary process that allows tumor to escape normal mechanisms that would control it, normal processes, physiological processes, but also would just allow these tumors to grow. So we know that many tumors overexpress PD-L1 and PD-1, and in doing so, they also have gained the ability and increased the ability to evade and to suppress the T-cell mediated anti-tumor response. So just going back, on the right you can see some examples of the drugs that have been either approved already by FDA for certain indications or being studied. So we can see, we know about Yervoy for CTLA-4 that was approved for the treatment of melanoma. This was one of the early examples, and now there are multiple molecules that have been FDA approved that are anti-PD-1 or anti-PD-L1, and both nivolumab or Opdivo and pembrolizumab or Keytruda gained a lot of notoriety. And there are companion diagnostic tests that are associated with those drugs. So I wanted to sort of focus on biomarkers more than on biology of immune evasion, and just give you, share with you certain, certain experience and knowledge that we've gained working with developing tests and also working with our oncology and pathology clients, but also with pharmaceutical companies in utilizing biomarkers. So PD-L1, it's just old immunohistochemistry approach, of course. I mean, there is the whole technique has not been revolutionized, but as we know, the immunohistochemistry at best is semi-quantitative under the normal sort of daily circumstances in most of the either commercial settings or even in the hospital labs. PD-L1 poses testing, and PD-1 testing, among other biomarkers, presents sort of the new level of challenge and complexity. Here I just give two examples of staining with anti-PD-L1 antibody in melanoma tissue tumor, and on the right in non-small cell lung cancer. Like many other tests, there are degrees of variability, and as I refer to it as shades of PD-L1, and complexity of really assessing the positivity in the tumor tissue. So PD-L1, many antibodies that are on the market are very robust antibodies, but they all have different staining characteristics to some extent, and I'm going to go into some comparative analysis of those clones actually later. But here what I wanted to point out is the challenges remain the same. It's semi-quantitative. There are different degrees of intensity. Here in non-small cell lung, you can see that there are some tumor cells that are very dark and bright and some very weak. The staining can be complete membranous, but can be also only partially membranous. There is an element of cytoplasmic staining along with the linear membrane staining. So what is important? How do we score this biomarker? Scoring in melanoma is complex because of the melanin pigment, and certain artifacts and presence of melanin can be very deceptive and can actually mimic the membranous deposition of the chromogen in anti-PD-L1 staining. So in that respect, from our experience, comparison with the isotype control is absolutely essential because melanin can present as a linear membranous staining also. So this is just the beginning of where complexity starts. But the question as it pertains to testing the looking at PD-L1 as the biomarker, and of course the question that has been raised now by many pharmaceutical companies and NIH is, can it be standardized, and how do we sort of proceed with that? And that question was unavoidable because on the market there are many antibody clones with various performance characteristics. The hospitals and different labs use different platforms from DACA, Ventana, Leica, etc., which also can produce different variable results, as we know. Also because different tumor types can have different very variable biomarker expression. And so, is it really cell surface expression that is important? Do other patterns like cytoplasmic expression carry any significance in certain tumor types, solid versus hematologic malignancies? What about expression of PD-1 and PD-L1 on infiltrating immune cells? How significant is that? Should we be scoring that? So all of those questions we are starting to answer, but I think the advent of companion diagnostics in immunohistochemistry was exactly precipitated by all of those complexities. In addition, pre-analytical variables. Of course, we all know how much fixation can affect the epitope recognition, and so this is something that can have a profound effect on the outcome, on the result of the test. And then lastly, the scoring algorithms. I touched upon the question of what is more important, or both, expression of PD-L1 in tumor or in tumor infiltrating lymphocytes or other immune cells, and what about the pattern and intensity. So all of those questions are still out there. Some of them have been answered. So how did we, and I'm going to sort of try to give some of the data or summarize the data that is out there, accessible, but also share our experience on how we tackled PD-L1 testing. Because in reality, now looking at this table, you can see that there are various, we already here listed 3 PharmDx or companion diagnostic tests. So there are choices. So how do we correctly select the test? How do we correctly really deliver the result and how do we correctly utilize the knowledge that have been already collected in order to really better and have an impact on patient care. So I started first, in the first row, I actually listed the LDT, or lab developed test. So this is based on the SP263 clones. So before companion diagnostics have been approved, we made the decision to, knowing what was going on in the field and that some of those companion diagnostics have been in the works, we actually started developing LDTs with various clones because we knew that they are likely to produce slightly different results. We started certain research collaborations trying to understand how these different clones perform actually in order to understand, which would be most appropriate for certain tests or testing certain tumor types. So we still have SP263 LDT because it's a very robust test. It is CLIA validated. So as a lab, knowing what the CAP and CLIA requirements are, it is possible to take RUO, research use antibody, and bring it into the realm of the clinical testing by just, doing these necessary validation protocols. And so after validating this test, then we as a lab have a freedom of offering it for clinical testing on different tumor types. So that sort of allows certain freedom of having the test that is not linked to specific drug, is not limiting on what tumor type, but still can give you the information, or give the clinicians and pathologists information on what is the PD-L1 expression in tumor types that still don't have companion diagnostics associated with it. I think it is very important because because we know about the off-label use drugs that are used after patients fail primary and secondary line of therapies. So many oncologists would choose to put patients on certain experimental protocols. But again, having the sense, and again, this is sort of course, this is the clinical decisions that oncologists and pathologists make, having a more educated decision about where the patient may potentially benefit from immuno-oncology drugs by testing the expression of PD-L1 or other biomarkers. So that's an example of taking the research use antibody or clinical even IVD antibody and validating it and then offering it with the disclaimer that it is not an FDA-approved test, but it is a clinical testing nevertheless. And of course then there are many companion diagnostics. So I listed here the 3 other clones that are most commonly sort of utilized because these are the clones that are foundations for the existing companion diagnostic tests. SP142 is the most recent actually. It was developed for urothelial bladder cancers that are for the patients that are benefiting from treatment with Tecentriq. So the clone is associated with Ventana. The clone is also available for LDT. So it is possible to buy the companion diagnostic test and then run and report it based on the guidelines. And I'll go and talk more about that. But it is also possible to buy the same clone and develop it into LDT. So when we get urothelial bladder cancer for testing, we know that what is the highest regulatory test that is available for patients who are candidates for Tecentriq treatment is SP142 PharmDx from Ventana. So then that would be the appropriate test to use. But if the oncologist doesn't tell us about the clinical, about the drug, then it's the matter of communicating with the clinicians to really figure out what would be the most appropriate test and what is the treatment plans. So I think now as CROs, as a diagnostic lab, we are in the unique situation, or which becomes even more relevant, of actually being able to be the link between oncologist and the patient and the pharmaceutical companies and being able to actually find the best test that will benefit the patient. And some oncologists are aware, and not everyone currently is aware of all of these complexities that exist actually in the field. Another test, companion diagnostic from DACO, actually two. One is based on 22C3 clone and one on 28-8 clone. They are both have been already approved for non-small cell lung cancer, 28-8 also for melanoma, that for the patients that are treated with specific drugs, and the readout evaluation of the test is also very different. I put here the thresholds just to illustrate the point that what we call positive and negative with each companion diagnostic is actually different. So just to review a little bit or to give an overview of the differences between lab-developed tests, LDT, and PharmDx test, which is FDA approved or FDA cleared. So both, in order for these tests to be offered to the as a clinical tool, as a diagnostic tool, they have to satisfy certain validation parameters. They have to be clear, approved within the state where the lab is offering that test, and both of them are if they're offered as a clinical test. But only companion diagnostic is FDA approved, LDT is not. And in that respect, PharmDx presents a higher regulatory test and may be recognized by treating clinicians and by some insurance companies as a medical necessity, more likely than an LDT, with all of the consequences that we're not going to discuss within the scope of this presentation. And then what is really the practical difference between companion diagnostics and LDT? LDT, it's really the lab that works out the protocol, concentrations, all of the conditions that go into the reliable, reproducible, sensitive test. With PharmDx, all of that work has been done and is very, very precisely spelled out in the technical sheet, in the instructions for the use. So the protocol is fixed, the platform is defined, the clone is defined. It's usually sold as a bundle depending on the company, and again it can be different with its negative control, with isotype control, sometimes with the certain positive controls that are recommended. The main also difference of companion diagnostics test is that the tissue type or the tumor type that it is tested for is also defined. So that test that you run then is would be only interpreted correctly on a specific tumor type. So if it is approved for non-small cell lung cancer, all the data and this whole companion diagnostics is relevant for non-small cell lung cancer. And it means that even if in melanoma the protocol doesn't change, the interpretation and what we call tumor positive, how we call tumor positive or negative may be completely different. And that is based on sometimes retrospective and sometimes prospective data that, that the associated with that specific drug and that specific tumor type. So as I mentioned, the scoring guidelines are also very, very precise, differently from LDT, where no one tells us how to score the LDT, what to call positive. And we as a lab choose to report the sort of the absolute numbers, the raw data, percentages, and let the clinical oncologist make the decision what they think, whether those percentages are sufficient or insufficient for then treating the patient. And as I said, they're linked to the drug. So we'll speed up a little bit just to show you a few examples of where we compared how these four clones that I mentioned perform in non-small cell lung cancer. You can see here example, there are two non-small lung cancer cases, and they are stained with four different, with one LDT, SP263. At that point here, SP142 was still used as an LDT. and then 2 companion diagnostic tests. And you can see how variable the staining is actually in different tumors. So if we focus here on the brighter, the lower panel, so this sample is stained very brightly by Ventana sp263 LDT and by DACA 28-8, LDT, but has much weaker pattern and staining with 22C3 and sp142. So this just gives you a glimpse at the fact that, and these all 4 tests have been optimized for non-small cell lung cancer actually, and still they give very variable staining. And here on the graph you can see that 3 antibodies actually have much better correlation than the last one here actually in green, sp142. So this is the performance characteristics that of these antibodies, just pointing out, summarizing what you saw visually, that even though signal to noise is very high in all, very low in all of them, the sensitivity is very variable, dynamic range is very variable, intensity varies also, and so overall performance can be rated. And again, for non-small cell lung cancer, this is sort of my overall performance schema. Just to switch into hematologic malignancies, because this is the area of our intense focus and interest, and so I wanted to focus on diffuse large B-cell lymphomas, where in DLBCL and in other heme malignancies, immuno-oncology drugs are also becoming very relevant. To give you an example, and again, this was started as a scientific collaboration with USC, and what we started with clones that have been published, like this Abcam clone APR 1161, and then other clones that at that time we wanted to test, and now we expanded it into a more inclusive, larger study with all the clones. And again, it demonstrates to you that in DLBCL there is a similar pattern. So I'm showing 3 different cases here, and you can see staining patterns with 3 different antibodies, how variable the intensity and overall % positive tumor cells that we find. So it's actually quite dramatic that you can go from 60% positivity to 0 positivity with different clones. The nature and the root of this is not entirely clear to me. Why epitopes, again, the most plausible explanation is that epitopes recognized, the epitopes of the PD-L1 that is recognized by different antibody is different and fared differently through pre-analytical fixation and processing. But maybe there are other reasons like mutations within certain epitopes or certain parts of the protein. That's also a possibility, but has not been proven to be true for PD-L1 actually. And this is the summary of the result. You can see that, again, there is correlation between 2 clones that I just showed, but with one of them actually there is a much lower actually correlation again. And this is some data that we obtained correlating PD-L1 with PD-1 expression, and there is a trend for inverse correlation, which is relevant for checkpoint inhibitors for the treatment of lymphomas. But again, I wanted that data. I thought that data can be useful. So in our study, what we actually showed was that when we do staining with all 3 antibodies, even though the intensity may be different, but when we dichotomize cases, when we look at the overall survival based on PD-L1 expression, and we measure expression with 3 different clones, we still do get similar trends. But it is because all of these 3 clones, they did work. The statistical significance for these Kaplan-Meier curves is somewhat variable and different between different clones, and that is reflected. And the difference in survival is different, and that reflects probably how differently those antibodies perform in that specific disease. But what we saw in our cohort was what has been published for DLBCL, that's that higher DLBCL expression, which is in blue, confers lower overall survival in cases with DLBCL. And we did look at correlation of PD-L1 expression based on the 3 clones that I just showed, as you can see here in this heat map, with other genomic markers. We looked at mutations, we looked at variable markers, we did applied NGS, platform that we have, or test that we developed for lymphomas that has 220 genes on it, including TP53. We use the FISH and immunohistochemistry to look at also specific markers. So the correlation that we found to be true is that one of the markers particularly, and it is either TP53 mutations, losses, or overexpression by immunohistochemistry, correlated with the higher PD-L1 expression. So we know that P53 loss or mutations confers usually the higher risk or poor clinical outcomes in DLBCLs, and so it's not surprising that there was a correlation with the higher PD-L1 expression. So what about going beyond immunohistochemistry? And I'll just want to mention a few things. Of course, the biomarker field is much more complex. In addition to immunohistochemistry-based techniques, there is also an expansion of the genomic techniques, molecular, flow cytometry, etc. And so we, as a lab, also try to stay abreast and develop those tests. So in the flow, we did develop recently the Immuno-Oncology flow panel, and we do find it very important, and I will mention there is an evidence, of course, that monitoring various immune populations using flow cytometry, populations within either peripheral blood or infiltrating tumor cells, gives a lot of important information and assesses or predicts responses to various immuno-oncology drugs. So this is our formulation of which populations we can detect. And again, it has a collection of antibodies and targets certain markers that allow defining the memory effector cells, naive T-cell populations, and in addition T and B regulatory cells. And so this is, the whole field is now I think, accumulated much more knowledge, and there is immunotherapy trials network that already gave certain indications on what should be tested in order to monitor and evaluate the responses to various checkpoint inhibitors and immuno-oncology drugs. And so here just in examples that certain markers that the network actually is suggesting to be tested. And again, you can see if you look at what test to be used, it's a combination of immunohistochemistry, flow cytometry potentially, and molecular markers. So we talked about neoantigens, and this is the evolving field of measuring the mutational burden in tumors, ability to predict neoantigens. And so we as a lab also trying to develop those tests, and we do in the meantime also utilize the MMR immunohistochemistry panel to try to at least identify those tumors that have genomic instability in some subtypes of tumors, and we are combining this with other biomarkers that allow us sort of more comprehensive assessment of which tumors may respond to immuno-oncology drugs. So that's in the nutshell what I wanted to share and discuss today, but just the conclusions are here very general in a sense that, of course, I think it's important to understand that the whole field of immune suppression is complex, that there are multiple axes the tumor utilizes, so it's not just about PD-L1, not just about CTLA-4, but there are various mechanisms, and that we need to assess them comprehensively using combination of tests, and also that we need to understand if we are measuring specific biomarkers, I think we all have to develop appreciation for the complexity of such tests. And with this, we have a little bit of time left for questions, and I think both Kamala and I would be happy to take questions. And you can see on the slide the contact information if you want to email us some questions and follow up on this discussion. Thank you very much for your attention.
Thank you, Rita. That was excellent, and there was a lot of information in there. So you could have had 2 or 3 talks carved out of that. While we're waiting for people to type questions in, let me start with a couple of quick ones here. I heard there was some movement to get panels of markers on a test by the FDA. They could be from different companies representing different assays as well as different drugs. I'm curious what your, either you or Kamala or Rita, either one, if you'd like to take that.
So if I understood your question correctly, yes, I think that it is moving in that direction because I think there is an appreciation that it's going to be not just about one biomarker but multiple, and because there are several ongoing combination trials. And I think the fact that there are combination trials, there is a need for simultaneously assessing several targets. I know that we are participating at least in one combination biomarker reproducibility trial. It is done sort of by the same company, but I see that those alliances are forming, and I know that there will be a combination companion diagnostics, and I think every company will solve it differently in terms of the arrangements, the intellectual property, and so on. But it's definitely on the horizon.
And I have a follow-up question to that, which is if companies don't have a companion diagnostic, or for that matter, an LDT with the drugs that they introduce, will they be at a disadvantage?
I think it's a complex question. My personal feeling is that, of course, there are advantages to the use of companion diagnostics because it offers just higher degree of reproducibility, and it is, in that respect, a more reproducible and quantitative test. But I also think that it offers brings with it a lot of complexities because of how rigid those companion diagnostic tests are, and they require certain platforms that a lot of labs don't have. So I see that either at the end, companion diagnostics will be centralized. There will be few labs offering certain tests, and so labs will become more subspecialized or will just maintain their specialty portfolio, and there will be more send-outs from other labs, or that companion diagnostic will prove to be too costly and too prohibitive, and at the same time we will gain more knowledge and data and understand how to use other LDTs to replace the companion diagnostics. So there is initiative by NIH comparing all clones of PD-L1, for example, and pharma that is backing up those clones actually participate and contribute to that trial, but also to other initiatives. There are several actually besides the NIH one, and so I think what will emerge, a better understanding of how those tests compare. And I think it is important because at the end it is about patient and about patient selection, right? And at the end, we want to arrive at the correct selection of the test and also correct selection of patients for the drug treatment. So I think at the end we will all benefit from a more open market.
Excellent. I understand there are a couple of questions coming in, and I will yield the floor to Joanne.
Great, thank you. Yes, we have several questions. The first is, the talk seems to focus on a certain type of immunotherapy and the benefits of biomarkers. Could the speakers discuss the current promise and role of biomarkers in other forms of immunotherapy, such as CAR-T and TCR?
Yes, it's certainly a big field, and that is definitely deserves a separate webinar. I haven't even touched upon that. There are many ways to engage immune system, from vaccine, the whole cell vaccines to CAR T cells that are engineered to recognize certain tumor epitopes, and they're all effective in different ways. So I definitely did not mean to emphasize that the the biomarker field and the immuno-oncology antibody-based drugs, I was not even trying to compare those to other ways of stimulating the immune system. And I would be happy to make a review on that topic at a different time. But the point is well taken. There are many ways to use immune system to fight the tumor.
Thank you. Here's another question. Well, actually, a couple of people have asked if we can make the slides available. We like to do that along with the recording, and we make them available as a PDF, and so we can discuss that. We will be sending out a notice to everyone when the recording is posted, which would be sometime next week.
Thank you.
Sure. So the next question, does SP142 behave differently from other assays because of the antibody difference or due to the staining process difference? Do you know?
It's a good practical question. So I don't know the whole truth. I don't know all the reason why SP142 has slightly different staining patterns. I think the answer is complex because SP142 performs better on certain tumor types, giving better intensity, higher dynamic range than in other tumor types. So from our experience, we did notice that it's somewhat tumor dependent, and that emphasizes the need to re-optimize these tests on each tumor type, really, that one is testing. So that's sort of one point that I wanted to make. SP142 doesn't have a dramatically different protocol. It's done on the Ventana. We use it on the Ventana Roche. It's a Ventana Roche antibody. We use it on the Ventana system first when we developed the LDT. And when we compare it to SP263, the protocol is actually very similar that we were using for LDTs between SP142 and SP263. So from that in that respect, I think it's more about the specific epitope that is recognized by this specific clone and how this epitope is expressed by different tumor types. Could it be reflecting certain mutations within one tumor type and not another, or just the intensity of expression or how the protein is processed in different sort of cell types? That would be my guess why we see such a different result. I don't think it is just a technical difference.
Great. We have one more question, and we can go on for a couple more minutes. If other folks have questions, feel free to enter them. Is it possible to detect PD-L1 expression levels on the circulating tumor cells for companion diagnostics?
I would say yes. We don't have. Currently we are developing the CTC technology and tests, but we have not launched them clinically yet. As based on the technologies that are available out there, most of them are based on either capturing or characterizing CTCs with the antibody. So I don't see why not. I think it would work quite well.
Beautiful. Well, that takes care of all the questions we have from our audience. Tom, I'm going to let you conclude. Thank you, everyone. This has been a really terrific program.
Thank you. Thank you for the questions and for attention of everyone who I don't see.
And I'd like to thank the speakers and thank you, Joanne, for hosting us. Very, very kind and generous of you. So I appreciate that. And I will just close out with saying, please, you're all welcome to come and register for the Companion Diagnostic event. You will see the link when the slides go out, and you can also find it simply at companiondiagnostics2016.com. So please, we look forward to hearing from you, and thank you for attending today. Joanne, any final comments?
No, I couldn't say it better myself. Thank you, everyone. We'll see you soon. Take care.
Thank you.
Thank you.